Hello, my name is Javier Sánchez Utgés, and I will briefly tell you about my latest work on LIGYSIS, a resource for the analysis of ligand binding sites, which I will be presenting a poster about in Montreal on session poster C, taking place on Monday, 15th of July. Small molecules ligands play an important role in protein function, acting as cofactors, substrates, inhibitors, or modulators. Identifying where ligands bind is key to understanding protein function and is also relevant for drug development. It is often the case that the active or functional site of the protein is known, but as you can see, ligands bind to multiple locations on the protein surface. The functional significance of these sites is unclear, and they may be functionally relevant or could identify previously unknown allosteric or other functionally important sites worthy of experimental investigation. To tackle this issue, we analyze all relevant interactions with the target protein to rank the binding sites on the protein surface based on a functional score. The top scoring hits can then be prioritized in a downstream analysis. To see more information about this, you can check the paper which we published in Communications Biology very recently. Here, we are looking at the structural superposition of 2,500 ligands binding to the same protein. All these ligands do not bind the protein at the same time, but the data come from hundreds of different experimentally determined three-dimensional structures. The first step to start undoing this complex network of protein-ligand interactions is defining the ligand binding sites. We do this by clustering the ligands based on the interactions with the protein, and so all the ligands binding to the same site are colored in the same color. The binding site is defined as the residues on the protein binding to the ligands within a cluster. By leveraging the extensive amount of data on the Protein Data Bank Europe, we make use of thousands of proteins comprising tens of thousands of ligand binding sites. Hierarchical clustering can be used to group these binding sites using their solvent accessibility profile. This is whether a ligand site is buried in the protein core or accessible on the protein surface. 4 clear clusters are obtained this way. These 4 groups differ significantly in accessibility and conservation, but most importantly, they differ in functional enrichment. A ligand site in cluster 1 is 50 times more likely to be functional than a site in cluster 4. Using this information, a score can be computed that allows for the ranking of the binding sites on a protein in terms of likelihood of function. This ranking can then be used to prioritize the top scoring sites for lead development in drug discovery. The LIGYSIS dataset comprises 30,000 proteins with 120,000 structures and 200,000 bound ligands. To facilitate access and interactive exploration of this data, I am developing the LIGYSIS web server. LIGYSIS is a Python Flask web application using JavaScript for the user interface, Bootstrap for stylings, and 3DMold.js for protein visualization. This is how LIGYSIS currently looks. In here we are looking at 103 ligands clustered on 8 different binding sites from 84 structures of bovine CAMP dependent kinase. The main panel in the center is the 3DMold.js structure viewer, which we can use to explore the binding sites, the residues that form them, the side chains, and their labels. We can also explore the residues at an individual level on the right side. These residues will change when we click or hover on a binding site on the left, either on the plot, interactive graph, or on the table. We can display the ligands found on each site. Remember, the sites come from clustering ligands based on their interactions with the protein. We can also display the water molecules, although might not be the most useful thing to do. And then on the tables we have all sorts of metrics and information on the left about the sites, average statistics such as average accessibility, divergence score, missense enrichment score, size, the cluster, defined by their solvent accessibility, and the functional score that we can use to rank them based on likelihood of function. On the right side, we have the uniprot residue number, multiple alignment column, divergence, and other data. We can change the data displayed on the plot and therefore explore in more detail the complexity of this data. And also we have hovering labels such as the structure, chain, residue number, amino acid, and atom that we are hovering on. I wanted to finish by thanking everyone that has helped in this research, especially Professor Jeff Barton, my supervisor, Dr. Stuart McGowan, Callum Ives, and Professor David Coase. Thank you very much for your attention. If you're interested in LIGYSIS, please come to my poster or contact me online. And I hope to see you very soon on ISMB in Montreal. Goodbye.